Hey everybody, welcome back. I hope you enjoyed your second lecture exam on the nervous system. There are two generally recognized control systems in the human body. One is the nervous system. We are now going to take a look at the other control system in these series of mini lectures, and that control system is called the endocrine system. To start off, we're going to learn about hormones and their signaling mechanisms. The endocrine and nervous systems together are considered the two major control systems of the human body. These are two systems which are going to work together to maintain homeostasis in our body. Now, the endocrine system is going to be for the release of hormones into bloodstream that travels throughout the body. In the endocrine system, the hormones are released into the bloodstream because the target is usually far from the site of hormone synthesis. These hormones are going to travel through the bloodstream and they're going to reach their target cells. They're going to bind to receptors that are located either on the target cell or within the target cell. And again, these targets are going to be cells located throughout the body. Because it takes time for these hormones to be generated, to circulate through the bloodstream and reach their target cell, the results of the endocrine system control may take hours. But the effects of the endocrine system do last longer in comparison to the nervous system. It is true that in the nervous system, there are certain cells that can release hormones into the blood. For example, one that comes to mind are the chromaffin cells found in the adrenal medulla. These release norepinephrine and epinephrine. But for the most part, the cells in our nervous system are neurons. They're going to release neurotransmitters. These neurotransmitters are not going to travel very far. They're going to travel over very short distances to either excite or inhibit another neuron, a muscle cell, or a glandular cell. And because they don't have to travel very far, they get results in milliseconds. However, there is a very brief duration of effects. The effects of the nervous system don't last very long. This slide just shows the general functions of hormones and what the endocrine tissues are. If you take a look at the organs that are in caps lock, like the pineal gland, the pituitary gland, the thyroid gland, those ones which are shown in caps lock are considered to be endocrine organs because they produce and secrete a hormone. So they are part of the endocrine system. Now, we won't get into things like the stomach or the kidney or the small intestine. We are going to cover the endocrine portion of those organs when we take a look at things like digestion and we take a look at excretion. What we're mainly going to concentrate on are going to be things like the hypothalamus and the pituitary. We're going to look at the thyroid gland and the parathyroid gland. We'll take a look at the adrenals, and we'll discuss a little bit of the pancreas, but again, the pancreas will probably be a little bit more, we'll probably do a little bit on the pancreas when we get to the digestive system. These organs, through their secretion of hormones, helps regulate things like the composition of extracellular fluid and the volume of it. They help regulate metabolism. They help set our biological clock. They have really important roles in the response of cardiac and smooth muscle. They can regulate glandular secretions. They have immune functions. They play roles in growth and development, and they have essential roles in reproduction. You may have learned this in anatomy, but let's remind ourselves what kinds of glands we have in the body. An exocrine gland is a gland that secretes a product into a duct, and this duct empties into a body cavity, or it might open up onto the surface. Great examples of exocrine glands would be sweat glands, oil glands, mucus glands, the mammary gland, the wax ceruminous glands in our ear, and numerous digestive glands. In contrast, an endocrine gland secretes one thing. It secretes hormones. It secretes it directly into the bloodstream and does not use a duct. These are things like the pituitary gland, the thyroid gland, the parathyroid, the adrenals, the pineal. But there are other organs that secrete hormones as a secondary function, and they're listed here. Again, many of these we're going to take a look at when we get to a particular system. But we will come back and take a look at things like the hypothalamus because it's a very, very important endocrine gland. As a physiology student, you should understand the three basic kinds of interactions that we find between hormones. This is going to be a permissive effect, a synergistic effect, and an antagonistic effect. With permissive interactions, or the permissive effect, A second hormone is going to require the effects of the first in order for it to function. For example, in order for luteinizing hormone to work on oocyte ovulation, there has to be a certain level of estrogen. In a synergistic interaction, or synergistic effect, the two hormones have their own separate effects, but when you combine them together, you get an even bigger effect. So thyroid hormone can result in the breakdown of lipids, a phenomenon we call lipolysis. Epinephrine can also cause lipolysis, but when you put the two of them together, you get an even bigger effect on lipolysis. And then finally, you're probably more familiar with this one, the antagonistic interaction. In antagonistic effects, the two hormones have opposite effects to one another. A great example is insulin promotes the formation of glycogen by the liver, and glucagon stimulates glycogen breakdown by the liver. It is really, really important that we control the amount of hormones being secreted by an endocrine tissue. Hormone secretion can be regulated by signals from the nervous system, by chemical changes in our blood, and by other hormones. And there are two recognized feedback control mechanisms that are known to be involved in controlling hormone secretion. The most common is the negative feedback mechanism, whereas the other one is positive feedback. Now, in negative feedback, the decrease or increase in blood level of a hormone is reversed. Something goes on in the body that reverses the direction of the system and either decreases the production of a hormone until it comes down to normal level, or increases the level of a hormone until it comes up to its normal levels. Positive feedback control, the change produced by the hormone is going to cause more hormone to be released, which will cause more hormone to be released, which will cause more hormone to be released, etc., etc. The negative feedback control is more common. We'll see a couple examples of positive feedback control. Generally, positive feedback control we don't see a lot because if this feedback control mechanism gets out of control, it can be very dangerous. But I will say that, a failure in these control mechanisms can often result in either hyposecretion, which is under secretion or low secretion of a critical hormone, or it can lead to hypersecretion, the over-secretion of a hormone. And this can eventually result in a disorder or a disease. The last thing I'll say about these feedback controls is this. The opposite of a negative feedback control is not positive. The opposite of a negative feedback control is no feedback control. The opposite of positive feedback control, no feedback control. The most common control mechanism for regulating hormone levels and hormone secretion is that of negative feedback control. In negative feedback control, a particular signal is received in the body and it causes the body to reverse the direction of a system. Let's take a look at this figure we have here, which demonstrates negative feedback. We have a guy eating a sandwich. As a result of eating the sandwich, the levels of glucose in his blood are going to go up. And as long as the blood glucose levels remain within the normal range, everything's fine. It's when these blood glucose levels increase beyond that of normal, this increased or high blood glucose level is going to be detected by the beta cells of our pancreas. And our pancreas beta cells are going to produce and release insulin into the bloodstream. As that blood flows through the liver, the hepatocytes of our liver are going to get that message. And they are going to respond to the insulin by taking the excess glucose out of the bloodstream and storing it within themselves as glycogen. That is going to drop our blood glucose levels down. And that signal then is going to negatively feed back to the pancreas. That drop of blood glucose back into normal range is going to reverse the system, and our pancreas stops making insulin. So we return to homeostatic blood glucose level. We have received a signal and we have reversed the direction of the system. That is negative feedback. What is a little less common in terms of hormone control is positive feedback control. And this is because if positive feedback mechanisms get out of control, they can be very dangerous. In positive feedback control, the change produced by the hormone causes more hormone to be released. And the best example that we can give for this is either lactation or childbirth. In this picture, we have positive control positive feedback control controlling lactation. We have a picture of a child suckling at the breast. The sensory receptors in the skin of the breast detect this suckling and they send a message off to the hypothalamus. The hypothalamus is going to respond to this message by producing a hormone called oxytocin. Oxytocin is then going to be released, which is going to cause the breast tissue to be stimulated and to eject breast milk. That ejection of breast milk is going to cause more suckling from the baby. It's going to cause more sensation at the breast, more hypothalamic release of oxytocin, and et cetera, et cetera, et cetera. It is imperative that cells talk to one another within our body. And the way cells can communicate with each other can vary. It depends on if the cells are right next to each other or if one cell has to talk to another cell located at a distance. So this slide outlines the three major ways that cells can communicate with one another. In way number one, the cell communicates with its neighboring cell through something called a gap junction. This is the simplest form of communication. The gap junction are membrane proteins, and these two membrane proteins from adjacent cells will line up with each other, connect to each other, and in the middle of those gap junctions, there'll be a tunnel. So if two cells connect to each other via a gap junction, the inside of one cell is directly in communication with the inside of its neighboring cell. Way number two is communication can be through direct contact, that is an adhesion-based mechanism. For example, one cell expresses a cell surface receptor, the other cell next to it expresses a ligand, and the two neighboring cells physically interact with each other through this receptor-ligand connection. So both number one and number two deal with cells that are right next to each other. Number three deals with how cells communicate with one another at a distance. Cells can communicate with one another through the production of extracellular factors called signals. These signals are things like hormones, neurotransmitters, neuropeptides, or growth factors. They are made by the cell, they are secreted outside of the cell, and then these signals are either going to travel through the fluid of a tissue or they'll travel through the bloodstream to their distant target. Now, this mode of communication is called extracellular signaling, and we're going to concentrate on it in the rest of this lecture because this is the way hormones communicate, or this is the way hormones are used by cells to communicate with each other. In extracellular signaling, extracellular signals are produced and secreted by a cell into the external environment. And this signal could travel through a tissue. It could travel to a nearby tissue. It could enter into the bloodstream and travel to a tissue located clean at the other side of the body. But this signal is going to exert their effects by binding to the outside of a target cell or by entering into the target cell. And the ultimate goal is to affect the function of that target cell. Over here on the right-hand side, we have a picture of an extracellular signaling molecule binding the outside of a cell via a receptor protein. Now, this extracellular signaling molecule came from another cell. It was produced and secreted by another cell, either into the fluid of a tissue or into the bloodstream, and it has traveled a pretty good distance to get to this receptor protein. Once it binds this receptor protein, it's going to set up a series of intracellular signaling events, which we're going to cover in this lecture. But the downstream effect is that eventually some intracellular signaling proteins are made as a result of this extracellular signal binding a receptor. As a result of this intracellular signaling, something's going to happen inside the cell. An effector protein is going to be affected. Maybe the effector protein that's going to be affected is a metabolic enzyme and you get altered metabolism. Maybe these intracellular signaling proteins affect the function of a cytoskeletal protein and the cell changes its shape. But more often than not, what these intracellular signaling proteins are going to do is they are going to change the level of gene expression. And by changing the level of gene expression, maybe we make a brand new protein that changes the function of the cell. Or maybe we stop making a protein, which again changes the function of the cell. But ultimately, at the end of extracellular signaling,
our cell changes its function.
In extracellular signaling, the signal is secreted by a cell and it travels to a target cell. Once it gets to that target cell, it's going to bind to a receptor. Now that receptor could be either expressed on the outside of the cell or it can be expressed within the cell itself. But we call the signal when that is binding to the receptor, that signal is often called a ligand. If it binds on the outside surface of the cell, if it binds the receptor on the cell surface, then we're going to have to transduce that signal into a series of intracellular events, which is known as signal transduction. If it binds the receptor inside the cell, again, we will then take that signal and convert it into a series of intracellular things. And again, those intracellular things collectively is called signal transduction. Because the ultimate goal of signal transduction is to change the expression of a particular gene, and thereby, by changing protein levels, change the function of the cell. So in this picture, you can see that we have extracellular signals binding to cell surface receptors. You can see that it is a very, very specific thing. The circular signal binds the circular receptor, the square one binds the square one. And then you have a bunch of things happening inside the cell. So that a bunch of intracellular events happen, we end up changing gene expression, and we end up making more of this green protein, and the cell ends up changing its function. If those receptors are found inside the cell, it's not going to change. You're still going to ultimately affect the function of the cell.
Here are the 6 steps to extracellular signaling. Number 1, you have to make the signal. That signal can either be a growth factor or a hormone, maybe it's a neurotransmitter. Then you have to release that signal via exocytosis, otherwise known as secretion. In the 3rd step, that signal's got to travel to the target cell. That target cell could be local, maybe it's located within the same tissue, or maybe that target cell is located at a distance, in which case the signal's got to get through the bloodstream. Once it gets to the target cell has to detect the signal. And this is done by the binding of the signal to a receptor. This receptor could be located on the surface of the target cell, or maybe it's found inside the target cell. And then in step 5, what results of that binding is a change in target cell function. For example, maybe the cell in response changes its level of gene expression and protein production. And the last step of extracellular signaling is removing the signal and losing the effect it has on the target cell. There are 3 categories of extracellular signals. There are paracrine factors, which act locally. These are usually things like growth factors. There are neurotransmitters, which act locally. And then there are endocrine signals, which act at a distance. And endocrine signals are called hormones. Now, we won't talk much about growth factors and paracrine factors. We've already talked about neurotransmitters because you've just gone through an entire section on the nervous system. So for the remainder of this lecture, we're going to concentrate on hormones. A hormone-producing cell is often referred to as an endocrine cell. And hormones can use 3 mechanisms to signal. They can use autocrine signaling, paracrine signaling, and endocrine signaling. Let's take a look at this figure on the right-hand side because it shows endocrine signaling, autocrine, and paracrine signaling. The top portion of this figure is endocrine signaling. We have our endocrine cell releasing hormones into the bloodstream. These hormones travel via the bloodstream to distant target cells where they either bind to a cell surface receptor, as shown in this figure, or they enter into the cell to find their receptor inside the cell. The next part of this figure is paracrine signaling. We have the paracrine cell releasing hormones, and these hormones travel over a pretty short distance to a nearby target cell. Again, once they get to the nearby target cell, they could bind to a cell surface receptor, or they could go into the cell to find its receptor. And then finally, the bottom part of this figure is autocrine signaling. The cell releasing the hormone is going to be called the autocrine cell, and the hormones are circling back to bind receptors located on that same cell, or they will circle back to enter into that cell to find their receptors. We're not going to be taking a look at autocrine signaling and paracrine signaling outside of this slide. The remaining parts of this lecture are going to concentrate on endocrine signaling. Okay, I lied to you. We are going to take a little bit of a brief look at autocrine and paracrine signaling mechanisms, but only in these next two slides. Autocrine means self. Well, auto means self. So in autocrine signaling, you are acting on yourself. In autocrine signaling, the cell is going to respond to the hormone that it makes itself. This is also used by growth factors. You can see the cell up in the top figure releasing its contents. That content, be it a hormone or a growth factor, is circling back to find receptors on the surface of that same cell. Or again, in the case of hormones, it could find its receptor inside the cell. When I say the word paracrine, I want you to think local action. In paracrine signaling, the local signal, called a paracrine signal, acts on a neighboring cell, located maybe within the same tissue. So in paracrine signaling, the local cell responds to a hormone made by its neighbor. You can see here in this figure, the cell secreting the signal, let's make it a hormone. The hormone diffuses over to its neighboring cell and binds onto the neighboring cell through a receptor, or it can enter into the neighboring cell to find its receptor. This is used by hormones, it's also used by growth factors, and it's used by neurotransmitters. In the case of neurotransmitters, just make this secretory cell here on the left-hand side one neuron, make the space between the two cells the synaptic cleft, and make the adjacent target cell another neuron. All right, as I said in a previous slide, we're now going to leave autocrine and paracrine signaling behind, and we're going to concentrate on endocrine signaling. In other words, we're going to concentrate on how hormones work. Here is one of the simplest slides you will ever see out of me in this course. It describes to you the two types of hormones, and those two types are water-soluble and lipid-soluble. Each of these types of hormones are going to have their own way of signaling, and you are going to learn these mechanisms right now. There are several kinds of lipid-soluble hormones. The majority of our lipid-soluble hormones are going to be steroid hormones. These steroid hormones, or as they are known conventionally as steroids, are derived from a lipid called cholesterol. Cholesterol is made in the smooth endoplasmic reticulum of specific tissues, and then different functional groups will be attached to the cholesterol core in order to change it from cholesterol to a specific steroid hormone. For example, the steroid hormone testosterone will be made by the smooth endoplasmic reticulum in the cells of the testes. And in those cells, it will switch out specific functional groups of cholesterol, put on different functional groups, and make testosterone. In the adrenal cortex, cholesterol, the cells of the adrenal cortex will take cholesterol and modify it to produce cortisol. And here again, you can see the difference between testosterone, aldosterone, and cortisol. And you can see that it is the same fundamental structure with different functional groups added on. Thyroid hormones are going to be called T3 and T4. T3 is triiodothyronine. T4 is known as thyroxine. And to produce thyroid hormones, all our cells do is modify the amino acid tyrosine with additional tyrosine rings and iodines. So here is tyrosine on the left-hand side. Change tyrosine into T4, you add another tyrosine ring on, and then you add 1, 2, 3, 4 iodines on those two attached tyrosine rings. Turn it from tyrosine into T3. Again, we add on another tyrosine ring to this amino acid R group, and instead we add 1, 2, 3 iodines onto the tyrosine rings. Other lipid-soluble hormones will include retinoic acid. Retinoic acid is a lipid-soluble hormone derived from retinol, which comes from vitamin A. Retinoic acid is really important in regulating things like cell proliferation, differentiation, and even the death of many cell types. You may not realize it, but vitamin D3 acts as a lipid-soluble hormone. And then finally over here, we also have nitric oxide, which is going to be involved in things like vasodilation of blood vessels. Nitric oxide, not nitrous oxide. Nitrous oxide is what you put in your car to make it go faster.
The last class are called the eicosanoids, which are the prostaglandins and leukotrienes. Both of these are going to be derived from a fatty acid called arachidonic acid. Both act in the inflammatory reaction. For example, they can stimulate smooth muscles to contract. They could stimulate nerve cells so that they become more sensitive to pain chemicals. To make a steroid hormone, I've mentioned that you have to synthesize it from a cholesterol backbone. This is going to require a series of specific enzymatic reactions that's going to take off some of the functional groups of cholesterol and attach brand new functional groups. These enzymes are going to be specific for each steroid being made. They are going to be found in very specific cell types. For example, the enzymes for modifying cholesterol into cortisol are found in the cells of the adrenal cortex. The cells that would modify cholesterol into testosterone or estrogen are going to be found in things like the testes and the ovaries. When you are making a steroid hormone, it's important to realize that steroid hormones, like most lipid-soluble hormones, are not stored in the cell. Once they are formed, they are released by diffusion into the bloodstream. It's only the cholesterol backbone that can be stored in the cytoplasm of the cell. And I believe this is the same pretty much for most of your other lipid-soluble hormones like the thyroid hormones, the eicosanoids. You can't store them. They are made and immediately secreted into the bloodstream. Remember that hormones travel via the bloodstream to a distant target. So we have a problem when we have a lipid-soluble hormone because the blood plasma is primarily water. How can a hydrophobic hormone interact and travel within a hydrophilic environment? They do so by binding to carrier proteins. These carrier proteins can be very specific to a certain kind of hormone, or they can be just general carriers of lipid-soluble hormones. So for example, they might pick up any steroid hormone, or they might bind to any kind of thyroid hormone. A great example of this indiscriminate carrier is going to be serine albumin. Albumin is one of the plasma proteins made by our liver. It is a general carrier of a lipid-soluble hormone like a typical steroid hormone. The hormone becomes activated once it is released from this carrier protein to its target, and then that hormone can enter into the cell. So really what our body does is it maintains a balance of inactive hormones bound to albumin carriers or bound to protein carriers, and then activated unbound hormones that are then rapidly going to enter into the cell. The mechanism of a lipid-soluble hormone is called endogenous signaling because it happens within the cell. More specifically, it is called endogenous signaling because the receptor for this hormone is found within the cell. I think this is the easiest hormone signaling mechanism. You start off with that hormone traveling through the bloodstream bound to a transport protein, the one that looks like the little blue wrench. Once you arrive at the target tissue, the lipid-soluble hormone is released from the transport protein. And because it is a lipid-soluble hormone, and with very few exceptions, lipid-soluble hormones are small, it easily diffuses through the phospholipid bilayer and into the target cell. And the receptor is going to be located within the cell. The exception to this is going to be the large eicosanoids, which are going to be too big to diffuse. But for the most part, most lipid-soluble hormones are hydrophobic and very small, so they have no problem diffusing directly through the phospholipid bilayer of the cell, or through the cell's plasma membrane. The site of action for a lipid-soluble hormone is the nucleus. Why? It is going to change the expression of specific genes. The hormone is going to bind to its receptor either in the cytoplasm and move into the nucleus, or it will find its receptor in the nucleus. Either way, that hormone receptor complex is going to find itself in the nucleus, and it is going to bind directly to specific sequences of DNA called response elements. These response elements respond to the binding of this hormone receptor complex by changing the expression of a neighboring gene. These response elements are usually part of what we call enhancers. You remember enhancers? And these enhancers are then going to physically interact with the promoter of a gene and enhance the expression of that specific gene. You get enhanced transcription of that nearby gene. You get newly formed mRNA molecules, which then get translated into polypeptides. The polypeptides form into brand new proteins, and these new proteins ultimately alter the cell's activity.
There are numerous animations that are available on YouTube. All you have to do is type in lipid-soluble hormone and you'll come up with many of them. Here's one that I've got for you right here. This one has no sound. It has no closed captioning and no narration, but it does really show nicely the mechanism of lipid-soluble hormone signaling. And while you watch it, you can put your own narration in.
As I mentioned, if you're a lipid-soluble hormone, you want to cross the plasma membrane. You can do this easily and directly if you are small and hydrophobic. But some lipid-soluble hormones can't cross plasma membranes because they're too large. These are things like the prostaglandins and the leukotrienes. In order for them to act, their receptors have to be on the surface of the cell. So they are going to use a mechanism which is going to be similar to the one we're going to look at now. It's called the water-soluble hormone signaling mechanism. Before we take a look at this signaling mechanism, let's take a look at the kinds of water-soluble hormones there are. There are the amine hormones. These are the hormones which are derived from the amino acids tyrosine or tryptophan. Hormones which are derived from tryptophan, that's serotonin, and the hormones that are derived from tyrosine are known as the catecholamines. This is dopamine, norepinephrine, and epinephrine. And you're thinking, well, wait a minute, these are neurotransmitters. Sure, if they're secreted by a neuron and they act on a neighboring cell, they're called a neurotransmitter. If they're secreted by a cell and enter into the bloodstream, they're called a hormone. Small chains of amino acids that form peptides can function as hormones. These are called peptide hormones. Two great examples are going to be growth hormone and oxytocin. And then finally, you have very large proteins called protein hormones. These are going to be made of either one or multiple polypeptide chains that wrap around each other in a very specific three-dimensional shape. Great example of this is shown here. It's called insulin. Water-soluble hormones are going to be made and secreted using the same mechanism that regulates the secretion of any other secreted protein. So for example, they are made as precursors in the rough ER. They get modified in the rough ER. They get transported to the Golgi, where they're modified even more. And then they are what I call pruned, or they're trimmed of excess amino acids to give rise to that active hormone. They're packaged, they're secreted from the trans-Golgi network. In some cases, that pruning or trimming can actually happen once they are secreted from the Golgi. But either way, you are going to make it as an inactive precursor, and you are going to process it and make it into a smaller active hormone. The thing is, unlike lipid-soluble hormones, you can store the larger precursor protein in the cytoplasm until you need it. And then once you need it, a specific stimulus will trigger its secretion.
Water-soluble hormones can easily travel through the blood because they are water-soluble. They are hydrophilic. But the minute they get to their target cell, they have a problem because they can't enter into the cell. They cannot diffuse through the plasma membrane. Therefore, in order for them to affect the function of a cell, they have to bind cell surface receptors that are expressed on the surface of the cell. And in binding to that membrane receptor, then they can give rise to an intracellular response. If you go to YouTube and type in water-soluble hormones, you'll end up with numerous animations. Here are two specifically that are made by the publishing company McGraw-Hill. The typical mechanism used by most water-soluble hormones is called G-protein coupled receptor signaling. And I want to go over it over the next couple slides with you. In order to perform G-protein coupled receptor signaling, you need 5 components. You need the hormone, which is referred to as the first messenger. You need a receptor, which is known as the G protein coupled receptor. You need a G protein, which is going to partner up with it. You need an effector, which is going to partner up with the G protein. And that effector is going to produce a second messenger. Now, depending on the hormone and the G protein coupled receptor, your G protein can either be stimulatory or inhibitory. The effectors can differ. The two most common effectors used in G protein coupled receptor signaling is adenylate cyclase and phospholipase C. And there are two really well recognized second messengers that cells use. One is called cyclic AMP and the other one is known as calcium. But there are other second messengers such as cyclic GMP, which are seen in certain signaling mechanisms. Let's take a look at the signaling steps in G protein coupled receptor signaling, or you can short form it to G protein signaling. First, let's go over the components. We have our ligand. Our ligand is going to be the water-soluble hormone. This ligand is going to be specific to a receptor protein, shown here in light green. That's our G protein coupled receptor. The partner to this receptor is going to be this G protein shown here in light blue. And the partner to the G protein is going to be the effector protein right next to it. So ligand, receptor, G protein, effector. And here's how it works. Our ligand is going to bind on to the G protein coupled receptor and it's going to activate it. You can see in this picture it goes from light green over on the left-hand side to dark green over here on the right-hand side. That's an activated receptor. The minute you activate a G protein coupled receptor, it is going to bind and activate its G protein. That's what's happening right here. You can see the G protein now, instead of being light blue, is dark blue. And the minute you activate the G protein is going to leave its receptor, move through the membrane to bind onto its effector protein, and it's going to activate that effector protein. And again, you can see the effector goes from light pink to this dark red. I will remind you the most common effector found in cells using this signaling is called adenylate cyclase. In some textbooks, it's known as adenylyl cyclase. But there is another well-described effector called phospholipase C. We're going to see in a bit that phospholipase C will produce calcium as a second messenger. For now, we're going to continue on with the effector being adenylate cyclase, and it is going to produce cyclic AMP as its second messenger. Our activated effector, shown here, which has been activated by binding to its G protein. This effector is going to then synthesize a second messenger within the cell. The first messenger is the water-soluble hormone that cannot get into the cell, so it causes a bunch of signaling events in the plasma membrane to occur so that eventually a second messenger gets made within the cell. If your effector is adenylate cyclase, it is going to convert ATP into the second messenger cyclic AMP. The effector phospholipase C will eventually result in the release of calcium from intracellular stores, and calcium will behave like a second messenger. The second messenger acts as an intracellular signal within the cell to activate a series of enzymes called kinases. It is going to activate a kinase. That kinase is going to activate another kinase. That kinase will probably activate a third kinase. So the job of a second messenger is often to activate a cascade of kinases within the cell. Ultimately, this second messenger, through however it's going to work, is going to result in the change in gene expression and cell activity. A lot of the times in these G protein signaling mechanisms, the very last protein that gets effected or gets activated in this cascade is a transcription factor in the nucleus. It binds onto a promoter and it helps promote transcription. A new protein is made and cell activity or cell function then changes.
Okay, what the heck is a kinase cascade? Well, let's back up a minute. Let's define what a kinase is. A kinase is a protein. It is an enzyme that phosphorylates a target, and that target is usually another protein. What a kinase does is it binds and breaks ATP down into ADP and phosphate. And then that kinase is going to take the phosphate and attach it onto the target, which is usually, again, another protein. This attaching of a phosphate onto a target is called phosphorylation. When this target becomes phosphorylated, it is going to become activated. And a lot of the times, the activated target is another kinase. This second kinase will phosphorylate and activate a third target. If that's a kinase, that kinase will turn around and will phosphorylate and activate another target. And you can have a bunch of kinases all in a row, like 5 or 6 dominoes all in a row, and you basically knock one domino over and all the other ones knock over. But usually what happens is at the very end of this kinase cascade, there will be a protein that will get activated inside the nucleus by being phosphorylated. And it will bind onto and increase the expression of a neighboring gene. And that's what you can see here in this picture. The phosphorylated target that I showed, that's a transcription factor. And you can see that phosphate group that I circled. Now you can see here how this phosphorylated transcription factor binds to a very specific region of DNA, and it promotes the expression of a nearby gene. And you get a bunch of brand new mRNA being made. That mRNA is going to be translated into, eventually, proteins. So again, if I say kinase cascade, think of a row of about maybe 4 or 5 or 6 dominoes. The second messenger is going to knock the first domino over. The first domino knocks the second one, third one, fourth one, etc., etc. At the end of that domino string of all those falling dominoes, you will have ultimately gene expression changing and the cell's activity changing.
This slide is a simplified version of what a kinase cascade could look like. A kinase cascade is going to start off with a G protein coupled receptor. So here's our signaling molecule binding our G protein coupled receptor. This figure does not show the G protein and the effector. And this activated relay molecule is going to represent our second messenger. So I'll draw here second messenger. I'm trying to draw second there with my mouse. I'm going to erase that, give that up. That's our second messenger. Okay, our second messenger is going to bind and activate the first protein kinase. There it is, all nice and active. That activated protein kinase is going to bind to a second target, and it's going to activate that second kinase. That second kinase is then going to activate a third kinase. And this could go on for 1, 2, 3, 4, 5 more kinases. Usually your protein kinase cascades are 4 kinases, 3 to 4 kinases. But ultimately, the very last kinase in this cascade is going to bind to and activate some kind of protein that is going to give rise to a change in the response of the cell. This active protein may be a transcription factor. This activated transcription factor would then change a level of transcription, give rise maybe to the production of a new protein, and now your cell is going to change its function in response to the production of that new protein.
A really important thing about a kinase cascade is because it can amplify the hormone signal. Here we have 1 molecule of epinephrine, and that causes maybe the activation of 3 molecules of the effector, adenylate cyclase. And that one single triangle right there in the center, you can see right here, makes 5 molecules of cyclic AMP, and each of them start a kinase cascade. Well, this single kinase right here can activate 5 other downstream kinases. This one right here can activate another 5 downstream kinases. So that 1 molecule of epinephrine can result in the transcription of thousands and thousands of strands of mRNA. Our cells express numerous kinds of G proteins. G proteins are transmembrane proteins. They are located in the plasma membrane. They are classified either as stimulatory or GS proteins, or inhibitory or GI proteins. Here you have a picture of a GS protein. It actually crosses the plasma membrane 7 times, so it's a pretty big protein. G proteins that are called GS stimulatory proteins physically bind and activate the effector so that a second messenger is made. GI proteins are going to bind the effector and prevent them from making the second messenger. The best studied simulatory G signaling mechanism is epinephrine binding to a G protein coupled receptor known as the beta-2 adrenergic receptor. Let's pretend this one here in green, this receptor here in green, is your beta-2 adrenergic receptor. You're going to take the epinephrine and bind it, and you can see now, take a look what happens to this protein when it binds the hormone. See how it kind of went from light green to dark green? But look, it has changed its shape, hasn't it? This little divot in the bottom right-hand corner now allows it to interact with the GS protein, which is shown here in blue. This GS protein is made specifically of 3 subunits, alpha, beta, and gamma. But we don't care about the beta and the gamma yet, we just care about the alpha, because the subunit that's going to travel over and interact with the effector is going to be that alpha subunit. When the GS protein is activated by binding to the receptor, it is the alpha subunit that is going to move over and physically interact with the effector. Now, it can only do this if the alpha subunit is bound to a molecule of GTP. Because when it's bound to GTP, it has the correct physical shape. And you can see how this GTP bound alpha subunit now can fit in with the effector. Let's pretend it's adenylate cyclase. And it can activate that effector. And now this effector, now that it's on, can take ATP and convert it to cyclic AMP. And then that cyclic AMP will go off and start a kinase cascade. As long as the alpha subunit is in its GTP bound form, it can stimulate the effector. But here's the thing. That GTP will slowly be broken down to GDP. And the minute it is bound to GDP, the subunit, the alpha subunit, comes off of the effector, finds its gamma and beta partners, and hooks back up again. It is now inactive. Think of this conversion from GTP to GDP as being like an egg timer that's going on in the background. And the minute that egg timer counts down and the timer goes ding, the GS alpha subunit now has to return to the beta gamma partner, and that entire complex then is turned off. The system then shuts off. Here's a medical application that directly involves the G alpha subunit, and it's called cholera. The G alpha subunit gets locked into its GTP bound on state by the cholera toxin. And remember, as long as this subunit is in its GTP bound state, it can then move over and activate the adenylate cyclase. So what you do is you have a system that never shuts off. You have a constant activation of adenylate cyclase, an extremely large production of cyclic AMP, and the cells that are susceptible to this mechanism line the GI tract, and they are responsible for absorbing water. Because of the increased production of cyclic GMP and the resulting kinase cascade and the change in cellular activity, these cells lining the GI tract cannot absorb the water they need to, and you have significant diarrhea to the point that so much water can be lost from the body through this diarrhea that it can be fatal within hours. The GI protein activates the same way. So don't worry about the left-hand side. Take a look right here at the right-hand side of this figure, because that's the inhibitory portion. This G protein coupled receptor is going to interact with the alpha subunit of the GI protein. This alpha subunit is going to interact with the effector, same effector, but now this effector will not make cyclic AMP. So effectors are targets of both GS and GI proteins. It depends on what alpha subunit binds you. If it's the alpha subunit of a GS protein, I'm turned on as the effector, I make the second messenger. But if I am bound by an alpha subunit of a GI protein, I don't get turned on and I don't make that second messenger. How do second messengers like cyclic AMP function in cells? They function by turning on kinase cascades. Cyclic AMP specifically binds to a class of kinase called a cyclic AMP, dependent protein kinase. That's a mouthful. They are short formed as PKAs. So cyclic AMP right here is going to bind and activate a kinase, and I can put a little star there, meaning it's activated, called PKA. The PKA will then go and find another target kinase, phosphorylate it and turn it on. That kinase will then go off and find another target, phosphorylate it and turn it on. That's a kinase, et cetera, until ultimately the very last thing that is phosphorylated is in all likelihood a transcription factor found within the nucleus. And that transcription factor that's been phosphorylated can then promote the expression of its associated gene. And you get increased gene expression and you get altered cell activity. There are numerous forms of PKAs. Each isoform is going to be specific to a certain type of cell. So for example, in epinephrine signaling, the PKA is called glycogen phosphorylase kinase. And it actually leads to the breakdown of glycogen into glucose. But if you are in, for example, I think like a muscle cell, the target of PKA is called acetyl coenzyme A carboxylase. So your PKA is going to be specific to the hormone, and it's going to be specific to the cell. Let's take a look at another very popular second messenger. It's called calcium. Calcium is stored in the smooth endoplasmic reticulum in cells. It can be stored in other calcium storage vesicles as well. But how does it get released? Well, you're going to need an extracellular signal like a hormone. That hormone is then going to activate a GS protein, and the alpha subunit of that GS protein right here, there it is in green, is going to bind an effector called PLC, phospholipase C. Phospholipase C now is an activated effector. It is going to act on a very specific phospholipid in the plasma membrane that is called PIP2. And it's specifically going to break PIP2 down into two things. It's going to break PIP2 down into a molecule called DAG, diacylglycerol, and IP3, inositol triphosphate. DAG stays in the plasma membrane. You can see it's still stuck to the plasma membrane. There it is. But the IP3 is going to travel and bind onto calcium channels located in the smooth endoplasmic reticulum. So there's our IP3 binding to this ligand-gated calcium channel. Here comes the calcium, which is going to come out now and function as a second messenger. The target kinase of calcium is called PKC. The kinase, which is the target of cyclic AMP, is called PKA. The kinase, which is targeted by calcium, is called PKC. PKC happens to stand for calcium-dependent protein kinase. But that PKC, you can see here, which is targeted by calcium, will then go on and set up its kinase cascade. Each kinase cascade is going to be unique to the cell type. So the kind of cascade you would get in a muscle cell would be very different than the one you would get in an adipose cell or a liver cell or an intestinal cell. Is there more than one kind of hormone signaling mechanism? Yep. Are these signal mechanisms also used by other signals like growth factors and neurotransmitters? You better believe it. Are the intercellular events, i.e. the signal transduction events, unique for each of these signaling mechanisms? Yes. But can they interact with each other? Yes, they can. This very pretty and very terrifying picture shows some of the very well-studied signal transduction or intercellular signaling mechanisms, many of which involve the production of second messengers. So for example, here's one right here. Here is our G protein cyclic AMP PKA mechanism. Here, right here, is the PLC calcium mechanism. Here are a few other ones that I worked in when I was a researcher at UCLA. Notice they all come down to the nucleus, and notice they all result in the activation of transcription factors And eventually you will get increased gene expression. It is pretty terrifying. I can tell you that when I was in grad school, I used to hate this stuff. If you gave a talk later on in the evening and you said the words kinase or cascade or signal transduction, my eyes would close and I would drift off into sleep. I couldn't help it. So of course, because the universe is a very ironic place to be, what did I end up doing for my 14-year research career at UCLA? I worked in signal transduction. I worked specifically in this kinase cascade right here called the MAPK pathway. Look it up. It's one of the prototypical pathways. It is studied just as much as the G protein signaling mechanism. Of course, we don't have time to go into these other mechanisms. To be honest with you, you can spend your entire life studying one mechanism in one type of cell and learning how it affects cell activity. So this was just a brief overview. I want you guys to concentrate on G protein signaling and the two kinds of second messengers, cyclic AMP and calcium. The next mini lecture in this series actually gets into the endocrine organs. So now that we know how hormones can work, let's take a look at the cells that are going to produce these hormones. We're going to start off with the two master endocrine glands, the hypothalamus and the pituitary.